Shri Parasakthi Vidyalaya Cbse Class 9 Standard Subject Biology.
Good morning, my dear students. Next, cytoplasm. The main feature of cytoplasm. That is, cytoplasm present between cell membrane and nuclear membrane. Amorphous, translucent, and colloidal liquid consists of inorganic molecules, water, and cell organelles. Next, the function of cytoplasm. Site for various metabolic activities such as biosynthesis of protein, fatty acids. Next, helps in exchange of material between cell organelles. Next, endoplasmic reticulum. The structure of the endoplasmic reticulum, the top smooth endoplasmic reticulum and the left side middle nuclear envelope, then the left side nuclear pore. Here cisternal space and cisternae. Next, rough endoplasmic reticulum, ribosomes. Endoplasmic reticulum is an extensive network of intracellular membrane bound tubules, sacs and vesicles extending from nucleus to plasma membrane within the cytoplasm. In most eukaryotic cells, it occupies the major part of cell. These membranes are structurally similar to plasma membrane and are composed of lipids and proteins. That is, endoplasmic reticulum is easily visible in young and dividing cells in comparison to old cells. It is the largest membrane found in the eukaryotic cells. It is absent in prokaryotic cells such as bacteria. It is also absent in egg cells, embryonic cells, and RBCs of mammals. Types of endoplasmic reticulum. On the basis of the presence of or absence of ribosomes, endoplasmic reticulums are of two types, that is the rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum contains ribosomes attached to its outer surface and thus have a rough membrane. It is found more in the deeper junctions of cytoplasm near nucleus, where it can be seen linked to outer membrane of nuclear envelope. The rough endoplasmic reticulum is more developed in goblet cells, fibrous blast and plasma cells. The protein synthesis occurs at ribosomes. Hence, the main functions of rough endoplasmic reticulum is to synthesize and transport protein. Next, smooth endoplasmic reticulum. Smooth endoplasmic reticulum, there are no ribosomes found on their membranes and is found largely near the peripheral part of cytoplasm, where it is stuck to plasma membrane. They do not take part in protein synthesis because their membrane lack ribosomes. The main function of smooth endoplasmic reticulum is synthesis of fats and lipid molecules. These are found in liver cells, intestinal cells, adipose cells, and muscle cells. Next, functions of endoplasmic reticulum. First, it acts as a supporting skeletal framework for the cell and gives rigidity to the cell. Second, it works as a channel and helps in transportation of materials between various regions of cytoplasm and nucleus. It is often called as circulatory system of cell. Next, it divides the cytoplasm into various small compartments. It provides a large surface for various biochemical reactions to occur. The smooth endoplasmic reticulum helps in synthesis of lipids, that is glycolipids, phospholipids and cholesterol, which are used in synthesis of membranes. Next, smooth endoplasmic reticulum helps in deoxification process of various drugs in liver of vertebrates. The rough endoplasmic produces digestive enzymes of lysosomes. The smooth endoplasmic reticulum synthesize hormones like estrogen and testosterone. Estrogen is nothing but it is the female hormone, sex hormone. The testosterone is the male sex hormone. Next, ribosomes. Ribosomes is the protein factory of the cell. The ribosomes were first discovered by George Plade in 1955 with the help of an electron microscope in animal cells. Thus, they are also called as blade particles. These are dense spherical granule-like particles of about 150 to 350 angstrom in diameter. The cell may contain up to 500,000 of ribosomes. These are made up of ribonucleoproteins and thus also referred to as RNA particles. The size of ribosomes is determined by the speed with which they sediment in centrifugal field. The unit to measure the sedimentation coefficient in Schwarzschild. Ribosomes are found in both prokaryotic and eukaryotic cells except mammalian RBCs. In prokaryotic cells, they float freely in cytoplasm. In eukaryotic cells, they are found in cytoplasmic matrix and also found stuck to cytoplasmic surface of rough endoplasmic reticulum and nuclear envelope. They are also found in the matrix of mitochondria and the stroma of plastids. They contain RNA and proteins and are not bound by membrane. Next, functions of ribosomes. These are the regions for the synthesis of protein in a cell. These are referred to as a protein factories of the cell. Okay, thank you, students.